This is part 2 of the lecture on viruses. We kind of got cut off on this slide in part 1. Let me go ahead and make it so I can write notes on here. So mainly, I was just saying that when you compare DNA viruses and RNA viruses, the biggest difference is that RNA viruses mutate a lot more frequently. Sorry, I didn't mean to draw over that word, but mutations in RNA viruses occur more frequently than in DNA viruses. And when we get to this section on the Baltimore classification system, I will go over this more with you as far as the reason why. The RNA viruses definitely mutate more often than DNA viruses. Now, a virus has to attach to a host cell in order to begin the process of tricking the host cell into creating new viruses. This particular illustration is KSHV virion, which is a single virus particle. Hopefully I can remember. It stands for Kaposi's sarcoma associated herpes virus. So there are many different types of herpes viruses, or at least there are multiple types, and this is one of them. And where you may have heard of it before is that in patients who have, have lower immune system, most often because they have AIDS, they can get like these brown places on their skin. They don't look like moles, they're flat, but they're these brown places on their skin, So anyway, that wouldn't happen in a person with a healthy immune system. So the sarcoma is the brown places on the skin. But this is an illustration used to show you how this particular virion attaches to its host cell. Basically, there is a particle on the virion that is shaped to fit a particle in the plasma membrane of the host cell. So in this case, these little green ovals are the viral receptors in the envelope of this virus, because this virus has a capsid. This is the capsid. Looks like a hexagon, but remember that it's three-dimensional. So we call that icosahedral. So the capsid is icosahedral. You're going to see this word again. The capsid is icosahedral. And then outside the capsid, this virus has an envelope, which is composed of a phospholipid bilayer with embedded proteins. And one of those embedded proteins is the viral receptor that you see in green. That viral receptor attaches to the viral receptor that you see here in yellow on the host cell. And the viral receptor on the host cell is called the XCT receptor. And then there are two other molecules that come in and kind of assist with this attachment, and they are called CD98 and alpha-3 beta-1, and they just help, I guess, make the attachment stronger. Make the attachment stronger. Okay, this is a very complex shape. And it's a special kind of virus called a bacteriophage T4. It's the same virus that I showed you in the beginning that was attached to the E. coli. Oops, I went too far. It's this virus, okay? So that's a bacteriophage. A bacteriophage is any virus that targets bacteria. The host cell is a bacterium. Okay, now the shape of this virus is called the head and tail shape. It's a complex shape, but it's called the head and tail shape. The head is everything above this line, and then the tail is everything below. And it is a DNA virus. That's also something that we see. So the core, its nucleic acid core is DNA, and then it has a head and tail structure. Another virus called an adenovirus causes many common respiratory diseases. The common cold can be caused by an adenovirus. It is usually caused by a rhinovirus, but an adenovirus can cause the common cold symptoms. But adenoviruses do not have an envelope. All they have is a capsomere. This is an icosahedral shape. Which is hard. The way the capsomeres are arranged, it's really hard to know exactly what geometric shape it is, but we just call it icosahedral because it's kind of round. That these individual brown ovals are the capsomeres, and all together they form the capsid. So this one does not have an envelope. It simply has a capsid, DNA in the core, and glycoproteins or spikes. And these are the structures that attach to the host cell, is those glycoproteins or spikes. This is an illustration, and then also, I'm so sorry about that, my phone rang. But what we're looking at here are images of the adenovirus. There are many different strains, but this is just a typical image. And you can see the shape better in the image on the left, better than you could on this page. But that kind of shape is sort of geometric type of shape, but it's rounded. That's called an icosahedral. So it has an icosahedral capsid. And then inside it has double-stranded DNA. And then this is a micrograph that shows the adenoviruses clustered together. And the adenovirus causes human eye and respiratory infections and sometimes intestinal infections. It's very common. We don't have a vaccine for it. It's not the primary cause of the cold, but it can cause a person to get a cold. Okay, now we're looking at HIV simply because just to compare the shape of the HIV to the adenovirus. So HIV stands for human immunodeficiency virus. Which of course is the virus that causes AIDS. Acquired immunodeficiency disease. This is a retrovirus, and its nucleic acid core is RNA, and it also contains an enzyme called reverse transcriptase. In 111, this is what we learned. We learned that DNA codes for protein by first coding for a messenger RNA strand and then coding for the amino acids, the specific amino acids that make the protein. So step one is when DNA codes for RNA, and step 2 is when the RNA codes for the proteins. Step 1 is called transcription. And that process uses RNA polymerase to help the DNA to produce a molecule of messenger RNA. But what we're seeing in what happens in the HIV is that it's a retrovirus, so it does reverse transcription. The RNA in the HIV uses reverse transcriptase to produce DNA. And once that DNA is produced, then it can go through regular transcription and translation and produce a protein. But the first step, RNA converting to DNA, is done using that reverse transcriptase molecule. This one does have a capsid, and it is covered. That capsid is covered with a an envelope. And the glycoproteins or spikes that attach to the host cell are found within the viral envelope. Okay, so here we see images on the left of the rabies virus and on the right of the variola virus. Rabies virus causes rabies, variola virus causes smallpox. And this is a picture of a person with smallpox. You can see that the rabies virus has a capsid and it has an envelope, and inside of that capsid is single-stranded RNA. And the smallpox has a capsid and an envelope, but inside that capsid is double-stranded DNA. So which one of these would be more likely to, would have a higher or faster mutation rate? If you said the rabies virus, you would be correct, because RNA viruses usually mutate at a faster rate than DNA viruses do. Okay, now I'm not going to spend a lot of time on this because this is just showing you some classifications of viruses based on their nucleic acid core. And since we don't really use this classification anymore, you don't have to learn these examples or anything. Here, this is classification by structure. You don't have to learn the specific viruses, but I do want you to know what naked icosahedral looks like. And that was your adenovirus, naked icosahedral. And then enveloped icosahedral would be enveloped would be like HIV, and Epstein-Barr. You may not know this from the name of the virus, but it causes one of the diseases that it can cause. What we normally call it like mono, but it's mononucleosis, and it can last for several weeks. It's very, very contagious. So that is one that is enveloped and icosahedral. Then there are enveloped helical viruses. Influenza, which causes the flu, the mumps, the measles, the rabies. Let's go back to that rabies virus. Okay, so in here, a helix looks like that, kind of like DNA, right? So in here you can see that the virus is wrapped around like that. You know, it's a helical shape. The actual shape of the virus itself is a helical shape, and then it is enclosed by an envelope. Then we have naked helical viruses, and that's the tobacco mosaic virus is our main example of that. And they just don't have an envelope around them. It's just the helical shape without an envelope. And then we have complex shapes. Sometimes a combination of icosahedral and helical capsid structures. The head and tail